All right, I think we're ready. I'm very happy to introduce Gabor Gyülveszi from Rejuveron Life Sciences. Thank you so much for coming, and I apologize if I messed up your name. I have the same problem. But thank you so much, and thank you for your support. Please, go ahead.
Thank you, Martin. Good morning, everyone. Good. Okay. Good morning. It's my first time at ARDD, and I'm amazed to listen to so many exciting and motivating talks. And somehow it reminds me, I'm coming from the cancer immunotherapy field. This meeting reminds me to the early days of cancer immunotherapy. I remember 20 years ago when I went to my first cancer immunotherapy meeting. It was a small room, even smaller than this one. Many enthusiasts in the cancer immunotherapy field, and everybody talked about the power of the immune system, and nobody believed in the immune system. And look at it now. I mean, 20 years later, immunotherapy plays a vital role in the treatment of cancer, and I hope a similar path is ahead of us for longevity and age-related disorders. And actually, that's why we are working at Rejuveron as well, and I'm here to present on behalf of Rejuveron and then our portfolio companies. Okay. Not ideal. What is Rejuveron? Rejuveron is a biotech incubator and an investor firm. We are headquartered in Switzerland, and we are building a diversified portfolio, and we are investing into companies and developing companies from our incubator which are tackling age-related disorders with a high unmet need. We work together with outstanding scientists in the longevity space. We work with drug developers who can help us really execute on our mission. And we provide the financial means and the drug development knowledge as well internally to help those ideas become realities. When you look a little bit more closer, how we can help our portfolio companies or how we can really create an ideas. We have an incubator space in Zurich where we either come up with ideas and try to validate or devalidate those ones. And we created an internal knowledge of drug discovery experts and additional scientists who can help really execute on this task. Additionally, we have a state-of-the-art incubator space. We have lab space in Zurich where we can have our scientists doing the wet lab activities, and they can really generate data. Additionally, we provide the financial support to our portfolio companies. We are the early investors in those companies, and if those companies are a little more advanced, we are investors, and we help them really to execute and deliver on their promise. Additionally, in our incubator space, outside of the financial support, we provide additional layers of support as well, such as legal and administrative support, and we really make the life of our portfolio companies easier because they can focus only on one thing, which is important for us, is science and developing drugs for patients. And the last thing, and I'm sure that you agree with that as well, network is critical. It's critical in academia, it's critical in industry as well. And we have a very strong network in the biotech environment, in the investor community, and among academic scientists. And we can help our portfolio companies to tap into this network and bring in that knowledge from the external world and profit from that knowledge. So that makes us, taking together these factors, that makes us unique. As I mentioned, we have our facilities in Zurich, and this is just a quick snapshot on our facilities. We have labs in Zurich, we have meeting rooms, and we have all additional facilities which are needed to run a successful drug development exercise. We work together with partners such as Harvard University, IRB in Spain, and other hospitals in the US and in Europe to really capitalize on the knowledge which is out there, and as well tap into their knowledge to create companies, bring in founders, and help them to realize their dreams and hopefully treat patients. You're very familiar with this graph, the 12 hallmarks of aging. And I just want to highlight here where our companies play among those 12 hallmarks of aging. Our most advanced company, Endogena, which is developing first-in-class therapies for ophthalmology indications. Their major task is to reactivate dormant stem cells in a variety of organs in their lead program in the eye, and hopefully they can create healthy cells in the body in a variety of organs and replenish really those organs with healthy cells. Another company which is in the clinic, and I'm sure many of you heard the presentation on Monday afternoon, is Rejuveron Biomed. They are taking a very different approach. They take a combination of 2 approved drugs and they develop that for sarcopenia. And this is a very different approach because doing so, you offload a lot of development risk because you understand the safety profile, the DMPK properties, and among other things of those drugs. So you can execute faster, and you can provide, hopefully, benefit faster to those patients in need. Another company, which is actually coming from our incubator space, is Rejuveron Senescence Therapeutics. And as the name suggests, it's working in the senescence. And they try to remove senescent cells in a variety of indications. And their lead program actually is focusing on an oncology indication, triple-negative breast cancer. And I picked this company, and I will showcase you the scientific data from their program, from their lead program. Another company, which is in the preclinical phase still, is Telomere Therapeutics. And again, as the name suggests, it works on telomere shortening. And it picked a very rare indication, is dyskeratosis congenita, which is characterized by the premature shortening of telomeres. And again, I will tell you more about this company in a couple of minutes. We have 2 more companies in the neurodegenerative space, Boost Therapeutics and Vascular Therapeutics. They have different approaches, but their mission is really to provide benefit to patients with neurodegenerative diseases. Vascular Therapeutics tries to fix the leaky blood-brain barrier and restore the functionality of that barrier, while Boost Therapeutics tries to enhance the quality of synapses in the brain and provide benefit to the elderly. A quick snapshot on our portfolio and where the companies are. As mentioned, Endogena is the most advanced company in our portfolio. They are wrapping up their phase 1b/2a clinical trial. They will publish data, efficacy, and safety data in retinitis pigmentosa early next year. Rejuveron Biomed, again, you heard the presentation on the preclinical data on Monday. They are running a phase 1b clinical trial, while the other 4 companies, they are in earlier stages. 2 of them are focusing on preclinical development, while the 2 neurodegenerative-focused companies are even earlier stages. They are in the discovery phase, and they are trying to identify their lead programs. A couple more slides on Telomere Therapeutics. I don't think I have to tell you the relevance of telomeres. You know very well that telomeres are caps at the end of the chromosomes, and that each cell division, they get shorter and shorter, and this is a completely natural process, and without this telomere shortening, our body would be in trouble, and we don't really want to interfere with this natural process. However, there are certain indications, certain diseases, where this telomere shortening happens way faster and prematurely. One of these indications is dyskeratosis congenita. It's an ultra-rare disease. It affects somewhere 1 in 500,000 individuals in the US. And what happens here, there's a certain mutation in the telomere complex, and ultimately that affects the shortening, that affects the re-elongation of the telomere, and it gets faster, shorter than in healthy individuals. It's a disease when there's no treatment, there's only palliative care exists. It's characterized as a multi-system stem cell failure because the stem cell compartment is affected, because their renewal capacity is affected. So those patients, unfortunately, they develop anemia, myelodysplastic syndrome, bone marrow failure, and a variety of fibrotic diseases. When you look at the overall survival upon diagnosis, it's a very poor number, unfortunately. 20% of the patients survive 10 years after diagnosis. The promise of this company is to develop not only a palliative care for these patients, but really a cure. How do we want to achieve that? Actually, this is just a very high-level schematic representation of what happens on our chromosomes. In these patients, actually, in contrast to healthy individuals, because this telomere complex is dysfunctional, telomere shortening happens way faster, and ultimately it results in a disease stage, so the telomere gain, in contrast to the telomere loss, is lower, and the telomere's length cannot be replenished as it would happen in healthy individuals. And the company identified a target which can be used for therapeutic intervention, and this target is part of the telomere holoenzyme complex, and we identified a small molecule which efficiently and selectively binds this target in the complex, and upon binding, the complex becomes functional, and we can re-elongate diseased telomeres, shortened telomeres, to their natural length. And it's important to point out that we don't interfere with the activity of the telomerase enzyme, and we don't over-elongate telomeres, because that would be dangerous. We really bring back to the natural state of those shortened telomeres. And just to show you a couple of more data points here, when you take a genetic validation experiment, what you see here, it's a photo of the telomeres of a healthy donor. What you see here is high up. Telomeres are around 20 kb long. When you take a patient bone marrow and you run the very same experiment, you see that those telomeres are way shorter when it comes to the healthy individuals. When we knock out the target in that particular patient, cell population, we can re-elongate the telomeres to their natural length, as it's shown on the photo. We made another step. Instead of using genetic validation, we used a small molecule, which does a similar thing, again, corrects the activity of the enzyme. The same order, what you see here, a healthy donor, a diseased patient, and ultimately we took the very same disease pool and we added the small molecule, and what you can observe that those telomeres became elongated and they almost reached the length of the healthy telomeres. So we can show clearly in vitro model, and I will not show you, but we have done in vivo studies as well in mice when we engrafted those animals with diseased or healthy stem cells, and we can achieve the very same effect upon systemic treatment with our small molecule. To give you a feeling where we are with this program, we are close to nominate a development candidate early next year, which we believe can be used for clinical studies, and if all goes well, we can be in the clinic in early 2025. We are working with one of the most prominent KOLs in this space, who sees those patients regularly and daily, and working with him provides us an added benefit, because he can recruit patients way faster. We can really design a clinical execution plan, which hopefully in 3.5 years can result in the generation of a data set, which can be used for submission to the FDA, and we can get potentially accelerated approval for this drug. Our hope is not only provide kind of relief to those patients, temporary relief, but really want to cure them, because we have very high hopes that the data, what I showed you, can be translatable, and we can re-elongate those telomeres in patients. The next company where I want to spend a couple more minutes is Rejuveron Life Therapeutics. This is a company, again, from our incubator, and we worked here together with Professor Manuel Serrano from IRB in Spain, because he's a very well-known expert in the senescence space. And we built our company on one of his observations. And actually that observation was that senescent cells actually upregulate PDL2, which you might be familiar with that target because it's another interacting partner of PD-1, which is a prominent therapeutic agent in the cancer immunotherapy space. But nobody has realized so far why PDL2 is important and what the role of PDL2 in cancer. We built this company on this observation, and actually I want to walk you through how we think this PDL2 works in cancer. This is a very schematic representation of a tumor. The tumor is composed of a variety of cells. Here you just see tumor cells. When a tumor or a cancer patient is treated with standard of care, such as chemotherapy or a targeted agent, CDK4/6 inhibitors or others, actually what happens in the tumor microenvironment, many cancer cells, they die because of the treatment. Those are the small ones. But some cells survive, and actually some cancer cells become senescent, and they upregulate PDL2, and at the same time they start producing a variety of factors, senescence-associated factors, which ultimately dampen the effectiveness of the immune response. What happens afterwards, because of this extremely suppressive microenvironment, the tumor regrowth, and PDL2 actually will get upregulated not only on senescent cells, but on a variety of cancer cells as well. And this is an inhibitory signal, and together with these senescence-associated factors, it's efficiently inhibiting the activity of the immune system, and the tumor cannot be removed from the body by the immune system. What we are developing at this company is a PDL2 inhibitory antibody. We have done our homework, and we looked into a variety of in vitro, in vivo models, and patient-derived xenografts. What you see here is really that PDL2 at steady state is not overexpressed or expressed by cancer cells. But when we treat those cells with the standard of care, palbociclib or other targeted agents or chemotherapy, they significantly upregulate PDL2. When we run the same experiment in vivo in animals and we implant a given tumor cell line, that cell line doesn't express PDL2. And that's obvious when you look at the graph. PDL2 treatment alone doesn't do anything because PDL2 is not expressed or it has a very low expression pattern. When we treat that tumor cell line with a standard of care, it responds to it. It upregulates PDL2 as it's shown on the other side of the slide. And when we add additionally other PDL2 blocking antibody, actually it's a PDL2 knockout experiment. When we knock out PDL2 on those cells, you can clearly see the drastic effect because there's no PDL2 anymore there to protect the cancer, and the immune system can do its job. We repeated the experiment with our antibody. As I mentioned, the earlier data point was a genetic validation. It's a PYMT model, which is a breast cancer model, and we treated those mice with a standard of care and added our antibody to the mixture. What you see here again, this is the very same data set or very same result that I just showed you on the earlier slide, that only the combinatory treatment where you have the standard of care together with other PDL2 antibody shows a drastic effect on tumor growth. And actually, when you measure exactly the tumor volume and the tumor weight at the end of the experiment, you can clearly see that the tumor is non-existent in those animals. The immune system is capable of recognizing that tumor. There's no inhibitory signal present in the microenvironment, and the immune system, in combination with the work of CD8 and CD4 T cells, will destroy that tumor. We did one step more, and we validated our hypothesis in patient-derived xenografts as well. Again, what you observe here, you can appreciate that doxorubicin and palbociclib treatment result in an upregulation of PDL2, and when you characterize the number of cancer cells carrying PDL2, that's drastically increased compared to the non-treated sample. So we strongly believe that the observation from preclinical models can be translated into humans, and we plan to initiate a clinical trial in the near future. This is a timeline associated with this project. We are still in the IND enabling phase of the program, and hopefully in a year time, we can initiate a phase one clinical study and prove the proof of concept of our hypothesis. To wrap up, I just wanted to show you a couple of highlights from our portfolio companies and mention it that we are working on a variety of indications which are age-related, they are unmet needs, and we hope that together with our scientists, with KOLs, with our external expertise, and the financial support from Rejuveron, we can really create therapies which will provide benefit to elderly with a given disease. And I'm at the end of my presentation. Thank you for your presence and happy to take questions.
Thank you so much. We have time for one very quick question.
Question about the economics of the telomerase-related drug. You said that you have like 500,000 patients per person.
No.
1.5 million.
1.5 million,
so
yes.
Meaning around 600 patients in the US. How can you make it economical for the company?
Yeah, that's a relevant question. Again, it will depend on pricing as well, because if it provides a cure, obviously you can price it accordingly. And if it's a cure, this is not a short-term treatment. Very often cancer therapies are priced high, but they are used for a year or 2 years, unfortunately. If this is a long-term treatment, patients will take it for decades. So you can build your model and kind of simulate it accordingly. So it's a long-term treatment with a relatively high price. So ultimately it will become economically viable. And at the same time, because of such a rare indication, you can apply for certain designations from the FDA, a rare pediatric voucher as well, which can be commercialized potentially on the secondary market. So there are certain factors which will make this project economically viable.
All right, thank you so much, Gabor. That was really fantastic talk.
Thank you.
Thank you so much for your support.